Assalamualaikum warahmatullahi wabarakatuh. Sakuan.
Hello, Assalamualaikum Doktor.
Waalaikumsalam warahmatullahi wabarakatuh. The time now is 11:30, so we may start our meeting with Al-Fatihah.
Okay, so boleh start kan Doktor?
Boleh. The floor is yours.
Okay. Okay, Assalamualaikum and good morning to Dr. Amizon.
Morning.
My name is Muhammad Zakwan bin Zabi, and today I'm going to share about my research project, which is about small scale to pilot scale fermentation using bioreactor system from oil palm frond. Boleh dengar Doktor?
Boleh dengar. Jelas.
Alright. Okay, next introduction. Before I start into my topic, I just want to ask Doctor or anyone watching this. So basically, is it every day in our life we use plastic?
Yes.
Yes, of course. Even
Lot.
my son also use plastic, such as we buy water bottle, we buy stuff at the stall, we need plastic. So basically, we use plastic in our daily life. So as you can see in this slide, the global production of plastic from 1950 to 215 is increased every decade and reached 350 million ton at 2015. Because plastic has a various application to its properties, such as versatile, lightweight, flexible, and highly durable. So it is not surprised that this production will continue to increase until further to 2021. But we have to question ourselves, after we use the plastic, did we recycle or even reuse it? So based on this statistic that only 20% of the plastic waste were recycled and around 24% were being incinerated. Meanwhile, the rest is discarded and mostly will be discarded to the surface water, around 10,000 to 100,000 ton of plastic in the surface water. And then this number will continue to increase and expected to increase because plastic require high cost to recycle and be incinerated. Anyway, petrol-based plastic is non-biodegradable, which can cause harm to environment because it does not compose easily. It takes around thousands of years to decompose by natural microorganism, and when it decomposes, it releases a poisonous properties to the environment, which can cause the sea, the any ocean water will be disrupted. Next, so how to tackle this problem? So polyhydroxybutyrate, PHB, a bioplastic. PHB is a well-known member of polyester, polyhydroxyalkanoic acid, PHA, which can be produced by lignocellulosic biomass. So PHB is I can say it's a game changer to replace and has the potential to replace petrol-based plastic. So as you can see in this slide, the polyhydroxybutyrate market share by region 2018 is increased in Asia Pacific. It is not surprised because mainly China or India is the economic powerhouse in this region. While the other graphic, you can see that bioplastic and biopolymer market increase every year, which means that PHB has the potential to replace petrol-based plastic. Like you can see, the comparison of PHB and petrol-based plastic, which made from polypropylene, polyethylene terephthalate, low density and high density polypropylene. This petrol-based plastic we use in our daily life, such as the plastic bottle. So these mechanical properties such as tensile modulus, tensile strength, elevation at break, degree of crystallinity, melting temperature, glass transition temperature. If you compare each of these mechanical properties of PHB and polypropylene and the rest petrol-based plastic, you can see that PHB is comparable and potential to replace them as a bioplastic. So we're moving on to the what type of biomass to produce PHB. So a lot of researchers has made their studies with different type of biomass such as sugarcane bagasse and glycerol and the rest you can see in the slide in effort to replace petrochemical based plastic with PHB. Different type of biomass can produce different concentration of PHB because biomass has a structure of cellulose, hemicellulose, and lignin. So as you can see in this slide, the different type of biomass can produce different biopolymer concentration. And of course, the different mode of working volume play a role, but the difference is can be negligible. Okay, next. Composition of oil palm frond and other biomass. So as you can see in the title of the study, oil palm frond. So in this study we use oil palm frond, but why oil palm? Basically, if you ride in the highway or anywhere, you can see your left side or right side, you can see a lot of oil palm plantation. So oil palm frond can used to produce PHB, and we can compare the composition of oil palm frond and other biomass. So you can see in this table the composition of lignin, hemicellulose, and cellulose is not the lowest, but it's not among the highest. It's in between, in the middle, compared to sugarcane bagasse, wheat straw, cornstalk, and sunflower. So production of PHB from biomass is basically from hemicellulose and cellulose. So you can see hemicellulose and cellulose composition in this oil palm frond is among the highest compared to other biomass. Okay, next, the pre-treatment. So what is pre-treatment? Before the conversion of PHB from biomass occur, pre-treatment is needed to break the lignin structure to allow the enzymatic replication by the microorganism. From the graphic, you can see that the lignin is wrapping around the cellulose and hemicellulose. If the lignin does not break, the microorganism cannot convert cellulose and hemicellulose sugar into PHB, which is a biopolymer. So, pre-treatment. strategies. There's a lot of pre-treatment strategies that researcher has been used, such as ultrasound, extrusion, alkaline pretreatment, ionic liquid, and also the findings when using this pre-treatment strategy is different, such as if we using extrusion, the maximum glucose is 75%, xylose 49%, and combined sugar 61%, obtained at 75 rpm and 1 to 5 degrees Celsius. So in this study, we are using alkaline pretreatment because according to Mr. Tan et al. 2019 in his studies, he is using alkaline pretreatment, and he is also mentioned that alkaline pretreatment is the best for biomass that has high content of cellulose and hemicellulose. Next is enzyme saccharification. Enzyme saccharification takes place after the pretreatment of biomass. So enzyme saccharification is a process of hydrolysis and fermentation using microorganism of treated biomass. So many parameters is affected using this when enzyme saccharification process going, such as agitation speed, temperature, and pH. So let's take a look at these all parameters and variation in production of PHB. So firstly, let's take a look at the agitation speed on PHB production. A lot of researchers has verified that optimum agitation speed is around 200 rpm. This is because agitation speed, its role in this process is to increase mass and oxygen transfer by constant mixing. So if the speed is above 200, the microorganism could die because it moving too fast and the mass oxygen transfer decrease. It also happen if the speed is too low. So the optimum agitation speed is around 200 rpm. Next, the temperature of PHB production. A lot of researchers has used temperature variations such as from 25 to 40 degrees Celsius, and the optimum temperature among this study is 30 degrees Celsius because the temperature affect the activity of the microorganism, where if the temperature is too high, it will kill off the microorganism. If the temperature is too low, the activity will decrease, which means the PHB production will also decrease. Last but not least, the pH of PHB production. So the pH level is used to change it can change the characteristic of enzyme active site. If the pH is too high or too low, it can affect the activity of the enzyme. So basically, the optimum pH is at 7, which is a natural pH. Next, the type of microorganism used in PHB production. So there is a lot of PHB, there is a lot of microorganism used in this PHB production, such as Pseudomonas albuminans, Bacillus cephensis, Ralstonia litropha. But you can see that some microbial is highlighted in green color compared to the black color. The green color is basically green microbial, which can be found around us, such as in soil, water. So in this study, we are using the Ralstonia eutropha. Most researchers also use this microbial because it has capable of degrading a big amount of chlorinated aromatic compound and chemically associated pollutant. So basically, this particular microorganism is used specifically to produce biopolymer. You can see in this table, the researcher use Estonia eutropha to produce different type of biopolymer such as PHB, PHA, and the rest. Next, in this study, we are going to look at the methodology to produce PHB. Firstly, our raw material, oil palm frond, are collected at local market and then dried by using ovens at 60 degrees until it reaches constant weight to remove moisture. And then the OPF is grinded around 0.2 mm size by using grinder and stored at desiccator at room temperature. Okay, and then the pre-treatment of oil palm frond. As I said earlier, we are using alkaline pre-treatment. So the OPF are soaked in 2% weak solution of sodium hydroxide at 121 degrees for 30 minutes with the ratio 1 to 9 solid to liquid phase. And then after 30 minutes, the OPF are then washed using deionized water until pH neutral. So at this stage, the linkage between lignin, hemicellulose, and cellulose are broken down, which open the way for the microorganism convert sugar into PHB. Then the OPM is dry until it constant weight and store prior to use. Bacteria strain. We are using Ralstonia eutropha, which is the subculture in the triptic soy broth without dextrose or at 4 degrees Celsius under standard condition and subculture monthly. So this is the composition of PSB, which contain nutrient for the microorganism. So enzymatic saccharification. As I mentioned earlier, enzymatic circulation is process of hydrolysis and fermentation. So for the hydrolysis, the alkaline pre-treated OPF is formed at biomass level 2% in 20 ml of 50 mm citrate buffer containing 0.005 sodium azide. The sodium azide is to prevent microbial growth, while the citrate buffer is to maintain the pH, because pH will play a significant role in this process. So the pre-treated OPF is added to 100 ml elemental flask containing the enzyme solution equivalent to FU activity of 20. Okay, this one I took from the researcher Ganesh Desaratel, and the mixture was incubated for 24 hours at 50 degree and 150 RPM in rotary shaker. So this is the first step of hydrolysis. The second step of hydrolysis occurs after 24 hours of incubation. The sample were filtered to separate liquid and solid phase. The solid fraction was then re-inoculated into 20 ml of 50 mm citric buffer and incubated at 50 degrees under shifting condition with 150 RPM. So there are two steps of hydrolysis because according to the researcher, you can see in the bottom left, an improvement of 9% in the efficiency of enzymatic hydrolysis was observed in two-step hydrolysis reaction as compared to single-step hydrolysis. So the second part of enzymatic second reaction is the fermentation. The Rastona eutropha were pre-cultivated in TSP medium for 36 hours. The cell pellet are collected by centrifugation, washed twice with phosphate saline solution, and then 1% culture transfer to the production medium. The production medium of PHP production has this new composition with addition of biomass hydroxylate. So you can see in this table basically contains nutrients such as potassium, iron, or the microorganism to grow during the PHP production. So the medium, the bacterial cells are incubated at 50 degrees Celsius with shaking at 150 RPM for 48 hours before the determination of cell dry mass and PCB content in the cell. During this process, the oxygen transfer rate are monitored by using blue sense monitoring system. So how to determine the cell driving and PHP content? After the fermentation, the sample were taken at the end of the period and is centrifuged at, at 11,000 grams for 15 minutes at 4 degrees Celsius. The solid were washed with distilled water and centrifuged for 2 consecutive times. Dry weight measure are carried out by drying the solid at 50 degrees Celsius and cool in a desiccator until it reaches constant weight. So the pHb content and the composition in the lymphophilin cell are determined we are using gas chromatography. So this is all from me. Thank you, Doctor.
Okay, thank you so much to Mr. Zakwan for a very nice presentation. A lot of improvements that you have considered and embedded in your presentation from our weeks of discussion. So a very good improvement here, but still I got more comments and I would like to start with, let us start with the first slide.
First slide.
Okay, you have a very interesting slide here with the oil palm frond shown. It's a very nice picture. And take note that whenever you use any image, make sure that the image allowable to be copied. Yeah, if not, then you have to add your citation at the as a footnote to your slide, just to acknowledge the owner of the image. That's one thing. Secondly, of course, there is no page number. Definitely, we don't put page number on the first page of any presentation, but we do have to put page number for all of the presentation slides, starting from slide number 2. Can we go to slide number 2? You don't have the page number there, so make it very proper whenever you make any presentation. Starting from today, now on, it has to have wajib, yeah, compulsory, the page number. Then it will help the audience to take note which slide they want to refer to when they want to ask questions to you. Yeah. Number 3, we should have a very nice citation. Okay, number 2 here, reference the image. Yes, I've mentioned it, slide number. Citation for the important points. For example, here, can we go to the next slide? Yeah, the same thing here. Yeah, if you don't go to the next slide, previous slide, and this one, you have a very important facts that you post on your slide. If you don't put any citation or referencing for those two images, I would assume that you plot this graph yourself, and those informations are collected by your own, yeah, not by others. I'm sure dekat image yang nombor 2 kanan tu ada dia punya It's very blur, tak nampak you punya citation code. But then you have to make it a practice, put your references as your footnote on your slide, saying that global plastic waste disposal ni dari mana, source Gaya kan, Gaya et al. It's very small. Cuba buat besar sikit warna hitam, tapi footnote lah, size footnote kat bawah. Then slide image number 2 tu daripada siapa tak tahu, adakah?
Ada.
Ada. So very small, yeah. It's very hard. Even you buat slideshow pun I tak nampak. If you go to previous slide, tak ada, betul?
Ada Gaya et al.
Ada juga Gaya et al, ya, nampak sikit. So this is quite better, ya. Tapi kalau if you want to make it more proper, standardized throughout the slide presentation, kalau footnote lah kat bawah tu. If not, saya tercari-cari kat mana benda ni. Ataupun you tambah teks lah, teks kecil right at the bottom of each images with a clearer punya ink. Ya, ini dia shade apa? Transparent punya style, kelabu-kelabu gitu kan, tak nampak. Ya, you can test it yourself bila you buat kita panggil apa? Preview, then you can see whether you can read it or not, ya. Okay, the fourth one, okay, third comment tadi, citation sama juga untuk, it's applicable to all the images for your total slides, ya. Ada banyak images lagi kan. If you have, then make sure that you do the same and make it standardized. Number 4, since this is a formal presentation and you want me to evaluate, and I will be giving you marks, yeah. So even most probably in the rubrics there it is not stated for any marks of presentation attire. So make it a practice whenever you make any presentation, since this is formal, not a normal meeting, pakai coat ke kat atas ke, nak pakai bow tie ke, ikut you lah, proper one. But normally bow tie pergi party kan, tapi not for presentation. So make it a proper attire. I'm afraid bila keluar nanti untuk you punya LI presentation ke apa-apa presentation to your superior or your managers, macam ni pakai t-shirt. So it's not proper, ya. So please make it as a practice. So that is number 4. Number 5, yeah, I do not know which slide, but I wrote here citation tadi tu. Saya rasa in one of the tables. Cuba you pergi tables on retreatment ke apa ke? Pergi lagi, next. Yes. Yeah, sini. Okay. If you notice, type of biomass tadi, Cotton Gin Das tu M. Tan et al. Tapi Court Stover, Wang. So the format of writing the references tu berbeza, ya. Wang, of course M tu dia punya first name, Tan tu family name, tapi Wang pun ada dia punya first name juga. Tapi normally we just put the family name Wang. So in this case Tan et al. Saja. So the consistency in citing the references must be only following one format, ya. Boleh?
Boleh.
Same goes kat sini, G Ramados lah, G Ravindran lah. Yang atas-atas tu format lain, bawah tu format lain. So that is my 5th comment. It's applicable to all, okay? Number 6, we have microorganisms, microbial. If you go to your table having types of microbial. Type of microbial. Ni. Normally when you want to write the microorganisms, it has to be written in italic. So Capria kecil, Capria vides, Necator, Pseudomonas, semua tu nama microorganism. They have to be written in italic, ya. In biological world, so mana-mana it's standardized seluruh dunia. When you write the microorganism scientific name, it has to be written in italic. Alright. Can we go to previous slide? Okay, another one, previous. Yes, this one, type of biomass. I assume when you write type of biomass. Apa dia?
Biomass.
Satu
Oh, this one.
Ya, ya, the one tadi tu betul. Tak apa, ada yang pink-pink tadi tu, pink-pink. No, the other one. Yang tadi tu, this
Oh.
one. Biomass, it's true, yeah. Since you are chemical engineering student, but you also learn bioprocess. So in chemical engineering, when we write biomass, EFB ke, open from ke, it's also biomass. But bila kita masuk biological pula, bioprocess, the microorganisms yang type of microbial tadi yang you letak tu, when we replicate it, produce it in a large scale, we also call it as biomass. The thing is, here, table Zakwan writes here, ada dua benda in one table, ya. You classify it as one. So I would like, it's not wrong, ya. For example, here, all palm from, of course, dia biomass yang chemical, cane, rice paddy, of course lah, lignocellulosic. In this case, kita nak differentiate bioprocess dengan chemical engineering. Satu yang tak ada melibatkan biological material. Of course, the all palm from is biomaterial, ya, tapi tak melibatkan microbe. But then the second one, you have streptomyces. Yeah, I'm not sure whether you know what it is. It's actually microorganisms. Of course, it's true. It's also biomass. I'm not sure whether when you relate it to PFB, in this article, Yongzo, Yongzo ni dua nama. Dia family name yang satu, Zo ke Yong ke? Please check. Yeah, sometimes
Alright.
ada orang family name nombor dua kan. So please check also on that. In that paper by Yongzo et al. In 2018, are they discussing on streptomyces as the microorganism used in the fermentation on what biomass? Macam paper lain dia guna OPF, kan? Tapi dia guna microbial tak? I just want to ask, Zahari dengan Yongzo ni. Ataupun macam mana?
Zahari tu dia memang pakai jus.
Dia pakai jus tapi ada guna microbe tak untuk consume the
Ada.
juice?
Ada.
Kan ada microbe kan? So you see there dalam table you, of course you are talking about the optimized condition of the agitation speed, but then classify that as satu centralized punya data. Table ni untuk OPF, tapi Yongzo guna on what? Juice ke? Biomass lain ke? Tadi Zahari guna tahulah microbe apa untuk OPF. Yongzo guna apa? Microbe ni untuk apa? Atas apa? Macam tu. So I got questions there.
Alright.
You see? So it makes me wonder apa yang berlaku kat sini, you see? Of course, that is not the issue here. The topic yang you nak present, effect of agitation speed. You bagi contoh je. But then it would be better if you could also classify, centralizekan dia.
Alright.
Nah, it's not wrong. Saya tak kata ni salah, ya. Tapi I just want to check your understanding. Supaya you faham dalam biomass tu ada juice tadi, of course dia still nak guna microbe dalam fermentation dia. Yang dalam Yongzo ni dia fermentation kot lah sebab dia ada microbe kan, tapi dia guna biomass apa. So ada persoalan lah di situ. It's actually I would like to know more when I see this, something like that. We have this discussion because I also want you to understand more supaya you nampak the overall overview. Okay, number 6, italic microorganism dah sebut. Number 7, okay, here with the same slide, Philip not mentioned. Maksudnya paper dia very simple. Dia tak mention pun biomass apa.
Dia tak mention.
Tak mention, so dia cakap apa?
Saya tak jumpa dia
Takkan dia
use
cakap dia buat
biomass.
operating dia atas air je? Tak cakap
Tak cakap tu saya.
Okay, in that case, because we want to train you to have the most recent data, yeah. In this case, this is also important. So that might not be data yang paling reliable kita nak follow lah. The rest tu we can, you see. But as an additional information, it's okay. Just that we wonder dia buat eksperimen atas apa. You see, to get this data. But anyway, dia punya RPM 125, the rest, 200, 175. 125 is quite low, yeah. It's very low. Normally 125 kita guna untuk anaerobic system. We don't want to allow very high oxygen consumption there. Yeah, just want to, I'm telling you based on knowledge, yeah. So that's the thing that I would like to share with you. Number 8, Streptomyces biomass saya dah sebut. Number 9, type of microbiota italic dah sebut. Number 10, soil and water, green microorganisms. Can we go to that table? Yeah, this one. It's very good that finally you also add more information on the green microbiome when we talk about as an alternative to the proposed microorganism. Apa tu nama?
Rastonia, Eutropha.
Rastonia, yeah. So we discussed on that in our previous discussion. That what about other microorganisms that we can easily extract ourselves without depending on buying from the bank, yeah, microbiome bank. So you got this finally, and this is the first time you present on this. I think I remember telling you maybe you want to back up it with the methodology as well. Did I say that in our previous discussion?
Backup methodology for green.
Okay, bermaksud, are you saying with the same microbiome tadi tu, we follow the same step? Hydrolysis, fermentation, macam tu?
Dia replace dengan microbiome lain.
Rastonia is also green there.
Sebab saya Google,
Rastonia bawah ni.
dia cakap can be found in soil and water.
Soil and water. So you boleh ambil Rastonia daripada soil and water, tak payah beli lah.
Macam tu. Tapi nak extract tu saya belum.
Beli? Sure?
Lagi.
Okay, alright, very good. So I'm happy. Akhirnya benda yang kita risau tak dapat beli tu, we can extract it ourselves from the soil. Yeah, it's quite interesting there. But then we were discussing on kalau kita ambil daripada nature, it can be mixed culture. Ya, tak ada specified Rastonia je. So I would like to suggest, please find out Rastonia green microbiome ni daripada soil ni. Please write down ya. Whether it's gram negative ke gram positive. And what is the size? What is the, not size, what is the shape of the microorganism tadi? This is considered as bacterial punya type ke? Is it bacterial?
Yes, bacterial. I think bacterial lah. Check lagi.
Okay, if you don't, if you are not sure, please take note the things that you can add. Sambil-sambil tu lah ya. For this one dah nampak cantik dah methodology. Cuma please find out kalau dapat kita extract sendiri, kita nak pastikan kalau mixed culture senang je. Kalau dia satu je spesifik kat sini, because from the soil dia tak akan ada mikrob ni je, dia ada mikrob lain ya. So kalau ada spesifik je benda tu, alhamdulillah, but specific soil, then we want to qualify benda tu betul ke tak by gram negatif ke gram positif. It depends kepada dia punya nature tu lah. Kalau dia gram negatif, kita follow gram negatif punya method untuk buktikan memang betul ini mikrob yang kita nak. Dan kalau shape dia rod, kita masuk mikroskop nampak rod, yes, betul lah. So inilah mikrob yang you guna untuk produce you punya PHB. Alright?
Contoh kalau saya jumpa mixed culture.
No, mixed culture is okay. It's okay. It's easier to have mixed culture rather than specific satu je, because normally from the nature you tak dapat lah satu je, susah. Memang susah nak dapat satu je. Alright?
Alright.
So mungkin Rastonia ni spesifik, dia dekat spesifik batu ke, dekat spesifik tanah ke, maybe ada. I don't know ya. So it's interestingly you can dig more information just for your knowledge, you nak faham Rastonia ni kan. Okay, number 11. Okay, from Ganesh, you were talking about two level of Two level of what?
Hydrolysis.
Two level of yes. Can you go to that slide? All right, this one, Ganesh, from Ganesh. You mentioned about the 9% increase of efficiency. My question would be, what is the final total efficiency from this two-level method? Stages method.
Maksudnya total efficiency after two-step hydrolysis compare dengan first step hydrolysis only.
Yeah, you mentioned about 9%, tapi total dia berapa? 80%. 9% full stage mungkin 50%. So, stage dia jadi 9%. So, now you know stage the final efficiency. Itu je. Can you have that in the article? Can you please just take note? Reply to the information? Boleh?
Boleh.
So just have that in the tahu lah ini our last meeting lah kot, no? For this semester, something like that. Unless we already start our work, then the meeting doesn't end lah. Then number 12, my third point, you were talking about slide after this word, fermentation. After methodology, fermentation. This one, 1% pre-culture. It's very shocking, 1% pre-culture is very, very low. So normally the common practice for you to put the inoculum in the production medium at least 10%. Bermaksud kata production medium tadi kan dia ada table sebelum-sebelum tu, ada yang buat in 3.5 liter, ada yang buat dalam 5 liter, ada yang buat dalam berapa mililiter. So when we want to add our pre-culture tadi, we call it inoculum. The common percentage 10 to 15%. 10 to 20%. 10, you see.
10.
But this one is 1, very low. So the fermentation might be very, very slow. So can you tengok balik, just take note, tengok balik yang dia tulis sebenarnya 1% tu. Saya mungkin maksud dia lain. When you write this way, I faham 1% pre-culture transferred to the production media, meaning katakan 1 liter lah, 1 liter you punya production volume, 1 liter. So 1% daripada 1 liter berapa?
1 lah, 0.001.
100 kali 1000 mililiter berapa? Kata dalam reaktor you ada volume 1000 mililiter Volume dia 1000 mililiter tapi size of the bioreactor tu mungkin 1.5 but you just put 1000 mililiter dalam tu. Then you want to add your pre-culture 1% daripada 1000 mililiter. So berapa mililiter you add?
100 ml.
Mana pula 1/100 kali 1000?
0.05. Salah sekejap.
Ni zaman ODM
10.
semua nak guna kalkulator. Pada guna otak je. 1/100 kali 1000. Berapa?
10
mililiter. It's very low, you see. So you bayangkan 1000 mililiter, you letak 10 mililiter. Then if you want to put that, the 10 mililiter must be very active punya Rastonia. Bukan yang tengah nak mati ke ataupun tengah stationary phase ke. Dia memang tengah betul-betul aktif exponential phase. You introduce to a new nutrients. Dia mungkin still very slow tapi dia taklah ada lag phase. The thing that I am talking to you yang you belajar masa chemical reaction 2 bioprocess. Growth phase punya graph. Ingat tak?
Ya, tinggi atas.
Lag, exponential, stationary, death phase. Kalau tak ingat, pergi balik buka nota, cari growth phase tu. So when we want to add our inoculum in your production, it must be at its exponential phase. Itu pun kita tak letak 1%. Commonly 10 hingga 20. Kita letak banyak. Sebab production ni kita nak cepat dan kita nak cheap. Maksudnya we want to cut cost. Kalau you letak very small, then there is like phase, dia guna electricity, guna energy to run your RPM, your agitator, electricity untuk set the temperature, cooling water, hot water, semua. Semua tu power consumption. Duk tunggu je dia duk adapt, duk let phase. Kan buang masa dan habiskan duit. So dalam industri, we want to cut that lag phase. So we want to put as optimized as possible the inoculum percentage. In this case, 1%. Please go back and check from whichever article. Ni pun tak letak, kena letak artikel apa. Dia punya recommended inoculum size berapa? Maybe dia maksud lain kot kat sini. Can you please check that?
Alright.
Not now lah. You pergi check lah bila-bila yang you free nanti check. Okay. My second last comment. This one as well. Next slide kot. Next slide. 50 degree C. Sebelum tu you cakap pasal temperature optimizer dalam
30.
berapa? 10
30 degree C.
Kan? 30. Tiba-tiba ni 50 lah pula kan. So tinggi jugalah temperature dia. So what you need to do is cari more information on Rastonia tadi. Orang lain duk study Rastonia optimize temperature dia berapa? Is it 30 ke 50? 50 ni selalu thermophilic punya microorganism yang memang suka temperature tinggi. Adakah Rastonia is a thermophilic microorganism? Thermo, nampak perkataan thermo tu maksudnya dia suka high heat, high temperature. High is considered like 50, 60, macam itulah. Itu pun sangat tinggi lah juga dikira walaupun thermophilic kan. Tapi depends lah on that microorganism. So the thing is we study on the microorganism lah. Mungkin that is the common characteristic about the microorganism, then we can accept it. But you mentioned about earlier literature, you 30, tiba-tiba methodology 50, so contradict. So what you need to do is tambah you punya literature lah. Ada orang find with this microorganism 30, it depends on the microorganism. It is not applicable to all microorganism. But finally you use Rastonia. Maybe ada beberapa literature lain yang duk cakap pasal Rastonia juga. So you found out memang 50. So valid lah dengan methodology yang you propose.
Okay.
You understand what I mean?
Yes.
Okay. So it's just a matter of validation and improving you punya literature to justify the method that you are proposing. Okay.
Alright.
The last one, finally last one. Still again here, kat sini ke? The next slide kot. Kat sini. The last sentence, the PHB content and composition in the lyophilized lyophilized cell. I think we mentioned about this in our previous discussion. Lyophilized, it's a lysis punya process, I guess. Lysis maksudnya dia kena break the cell to get the PHB. So I think I asked you earlier, Rastonia ni intracellular ke extracellular? Intra means PHB dalam cell. So you have to break it to extract the PHB. Kalau dia eat extracellular, selamat. Maksudnya dia produce dia punya PHB kat luar sel. So kita separate dia cell from the PHB. So daripada ayat ni, when I read this, I understand that kalau lyophilized ni dia kena melalui lysis dulu. You have to break it. So can you please find out and understand the nature of Rastonia, intracellular ke atau extracellular? Then understand the meaning of lysis tadi. Lyophilized dia apa benda? Yeah, so that you understand whatever that you write, mesti ada dia punya steps di sebalik ayat itu. Okay? Alright? So, gratefully, that is my last comment. Alright, congratulations. So, nampak some kind of improvement there. It would be very nice that we could try this in the lab. I'm not sure whether we can. So anyway, for the sake of the FYP, the 14 weeks that we have had, you have presented a very good progress. So congratulations. Even though those are new, baru teori kan, tapi betul dia punya practical belum lagi ya. Yang tu memang lagi demanding lah the effort from you because they are all very new to you, and also I've never produced PHP before. So gratefully I thank to you, of course, because you have put the effort here. I have the knowledge, but I've never tried producing my PHP, the PHP myself. Yeah, so congratulations. This is your proposal in producing PHP from all palm from. Yeah, so it's very good. Yeah. So my last word, again, thank you, and yeah. So we'll see again in a different discussion. Yang ni hari ni presentation evaluation saja. Okay?
Okay.
Do you have any last words?
Just nak cakap thank you to doctor for guiding me, and then for exposing me to different kind of parameters, new term in biological process, and everything lah.
Yeah. Okay. No problem. So it's also new to me, very new to you. So we have a very good cooperation there. I hope all the best to you. Yeah, Sakuan?
Okay.
I'm just waiting. The last step will be I'm waiting for your thesis. So please take note all the comments that I gave you. Of course, before you send me your thesis, you please make sure that you check your grammar consistently ya. Tak nak datang pada saya pening kepala tengok Allah ayat tunggal langgang ya. But so far reading your presentation pun saya tak boleh nak jangka lagi how you write your own thesis ya. Sometimes student bila dia tulis thesis dia ayat sedap sebab dia copy. So please do not copy directly. So please try to form your own sentence. It's a very good effort. But then when you try to write your own sentence, please check your grammar. So when it comes to me as a supervisor and also evaluator, we can focus more on the content rather than English, you see. So dia akan jadi demotivating sikit lah bila kita baca alamak tersangkut English. So tak apa sedap lah. So why we do that? Because we want you to be as our chemical engineering graduates from UiTM yang is well known to be using English all over, nearly 100% shown by your product. Bila you tulis ayat, you buat report, everything is consistent. Yeah, but you still got another semester to improve that. So please take that opportunity to improve whatever is before you graduate. Boleh ya, Zakuan? Alright. Thank you.